So today's video is going to focus on meiosis. Meiosis is going to be similar to mitosis, but not the same, obviously. It's going to involve sexual reproduction, not asexual reproduction. So for meiosis, we're trying to create change and trying to create different kinds of cells, as opposed to mitosis, where we were trying to have the cells be identical, exact clones of the original. Okay, so let's do a quick reminder about mitosis. Remember, mitosis makes two identical daughter cells, right? So we're making two identical daughter cells. So they both have the same amount of DNA. Remember, the cells are going to be that 2N diploid formation. So they're going to have chromosomes in pairs. There's going to be one division of the cells. And remember, this is for mitosis is for growth, for renewal or replacement, healing essentially, as well as asexual reproduction. So reproduction with no variation, essentially where you are creating a clone. Remember, mitosis had 4 parts. The cell cycle as a whole, remember, spent most of its time in interphase. And then mitosis, most of its time in interphase, doing its everyday stuff. And then mitosis was the PMAT portion. We went through the prophase, metaphase, anaphase, where they're pulling the sister chromatids apart. And then telophase, where we're going to reform the nucleus around the separated DNA. Chromatin will uncondense or uncoil, and those spindle fibers will go away. So we do have some organisms that do reproduce asexually. A few single-celled eukaryotes, so remember, have membrane-bound organelles, not talking about bacteria. Bacteria do, as a general rule, reproduce asexually with binary fission, but then they also have some other ways to incorporate genetic diversity into the organism. So some single-celled eukaryotes like yeast and certain protists, when we talk about kingdoms, that'll make more sense, but amoebas, paramecium, microscopic organisms. A few very simple multicellular eukaryotes, for example, a hydra, may also reproduce asexually. The question down there from Little Penguin, right? What are the disadvantages of asexual reproduction? What are the advantages? It's much faster, and it doesn't require a partner. Disadvantages, though, are that cloning part, that lack of genetic diversity. That lack of diversity can actually end up being a hindrance or a problem for the organism later. So organisms, for stability purposes, organisms, ecosystems, they like diversity. So we reproduce, as it says here, a complex multicellular organism. We reproduce by the joining of gametes, right? A sperm and an egg cell have to come together for us to be able to reproduce. And those gametes, those sperm and egg cells, can't be formed by mitosis. Remember, mitosis makes clones, genetic clones. So if I made genetic clones, then an egg cell would have 46 chromosomes. A sperm cell would have 46 chromosomes. Therefore, the new baby cell would have 92, and that would not work. That would result in the death of the organism. One extra chromosome can result in the death of the organism. So let's look at a picture of our chromosomes here so we can kind of get a feel of what's happening in the cell. We're going to use our cells as an example. It's by far usually the easiest. So the picture of the chromosomes here is called a karyotype, right? So this is this picture of the paired up chromosomes. And you'll see that each chromosome is in a pair, right? So I have 23 pairs. That's my 2N, right? That's my 2 times 23, right? Or my diploid number is 46, my 2 times 23. So these chromosomes are paired up. So one of these chromosomes came from mom, one came from dad. And that's the same for every pair, right? One came from mom, One came from dad. The chromosomes are going to be paired together and then numbered 1 through 22. And pair 23 is going to represent the sex chromosomes. So pair 23 is the sex chromosomes. If you remember from previous video, the rest of these are for somatic cells. So the rest of these pairs of chromosomes are called autosomes. The sex chromosomes are either going to be two X's or an X and a Y. This is a female karyotype, therefore an XX, meaning still got one of those X chromosomes from mom, one of those X chromosomes from dad. Male karyotypes will look the same. Again, this is a human male karyotype. 46 chromosomes or 23 pairs. They've all been paired up. My sex chromosomes are down at the bottom. You'll notice this time there's one X and one Y. And so that's those are the chromosomes that make the difference between male and female. This X chromosome actually had to come from mom, because remember, females are XX, so she cannot pass on a Y even if she wanted to. The Y chromosome had to come from dad, because remember, dad is XY, so he could have passed on a second X or he could have passed on the Y. So if you remember, we've talked about this vocabulary before. Those paired chromosomes, like you just saw on the karyotype, those are homologous chromosomes. Remember, homo means the same. So those chromosomes code for the same characteristics or the same characters. And you get a set from mom and a set from dad. Okay, so you can see in this cell example here, it's a 2N diploid, okay, meaning it has 4 chromosomes in total. So its haploid number would be 2, right? If it just had one of each instead of pairs of each. And those chromosomes are, for instance, these would be homologous chromosomes, and these short ones here would be homologous chromosomes. Let's say the blue is from dad and the red is from mom. So when you end up pairing them together, you've got a set from mom, a set from dad. Okay, so again, homologous chromosomes code for the same characteristics. They are normally set up like this in this single-stranded format until they need to be replicated. So whether you're going to do mitosis or meiosis, the DNA has to be copied, and that's when you get those double-stranded homologous chromosomes, is only if the DNA has been replicated. So one of the main goals of meiosis is going to be to reduce that chromosome number. We have to reduce the chromosome number by half. That way, when they come back together for fertilization, the zygote or the new combined cell has the appropriate number of chromosomes. So I need to go in the original mom cell that has 46, I need to reduce those chromosomes down now to 23 so that when the sperm and the egg cell come together, now I get 46 chromosomes and not 92. So let's look at what's happening overall in this life cycle of an organism that reproduces sexually. So we've got the gametes, right? I have an egg cell and I have a sperm cell. Fertilization occurs, which fertilization is just the two gametes coming together. So now I have the zygote, or that new fertilized cell that can duplicate. That cell will actually go through mitosis because the organism needs to grow. So it needs to produce more and more. Okay? And so this zygote has 2 copies of each chromosome. It's a diploid cell represented as 2N. Okay? When that organism is ready to reproduce, it will need to go through meiosis so that it can create gametes that can then go fertilize with other gametes. Okay, so let's look a little bit closer at meiosis. Meiosis is a form of reduction division. We're reducing those chromosomes from 2N to 1N, so going from diploid to haploid. This is going to take 2 cell divisions. So different than mitosis that only took 1 cell division, and it's going to make these gametes, and that's it. So how mitosis was for renewal and healing and growth, and asexual reproduction, meiosis is just for making gametes, making these sperm and egg cells. Okay, we are not going to get bogged down in the steps of meiosis. What we need to realize is that the steps of meiosis are PMAT again, and it just does PMAT 2 times. So there's a prophase 1, metaphase 1, anaphase 1, telophase 1, and then it starts over prophase 2, metaphase 2, anaphase 2, telophase 2. The end half of this, the 2 parts, those are going to be just like mitosis. I'm going to separate the sister chromatids there. Where the first part, the meiosis 1, is where I'm going to separate the homologous chromosomes. And we'll look at that a little bit closer on our next picture. But don't get, like I said, bogged down in this. Understand that I've got 2 cell divisions happening. Both time it divides, it goes through this PMAT process, prophase, metaphase, anaphase, telophase. So meiosis is going to have PMAT 1 and PMAT 2, where mitosis just has a single PMAT. Okay, so like we said before, meiosis has to go through DNA replication first. We can't just split the DNA. We actually end up making 4 cells here, so I need enough DNA to distribute it out to all 4 cells. So it's going to go through 2 divisions. So we're going to replicate the DNA in interphase like we did with mitosis. So DNA replication is occurring here in interphase. After the first PMAT, we've separated those homologous pairs. So you can see this happening over here on the right. Those homologous pairs have been separated. So what's happening here is I've reduced the chromosome number. So when those homologous chromosomes separate, the chromosome number becomes reduced. This is the division where we're going from that 2N to 1N. I know it doesn't look like it because it looks like we have these pairs now that are still there. So it looks like we should still be in pairs or diploids, but those are sister chromatids. Those are sister chromatids and they are replications. Okay, these are not unique chromosomes. These are the copies that are going to be separated out. So then the second division is going to separate the sister chromatids. So with meiosis, we're going to go through 2 divisions, which is going to result in 4 gamete cells. So I'm going to go from 2N, N. So with mitosis, I'm going to result in 4 genetically unique daughter cells. Mitosis gave me 2 genetically identical daughter cells. Meiosis will give me 4 genetically unique daughter cells. They are different from the parent and each other. And we'll look in a second about how they become different from each other if all I did was copy the DNA. So in getting ready for meiosis, like we said, first thing I need to do is duplicate the DNA. So in this example here, my diploid was a 2N cell. This cell, you'll notice, still diploid, still a 2N and telling you it has 6 chromosome cells, because those copies, they don't count the copies. We're only counting the unique chromosomes, not the copies. Okay, so I'm still at a diploid cell here that I'm going to divide. Okay, and then my first division of meiosis is going to separate those homologous pairs. So this is where I get that reduction in chromosome number. So if we look at our example here, I'm diploid at the top here. 2N, 4 chromosomes, pairs, diploid. They were then copied. So it's just double stranded. So remember, those copies aren't counting. When the division is all over, okay, you'll see I have 2 chromosomes that are copies. So now I'm down to an N. I've got that chromosome reduction down to those haploid cells. The second PMAT, the second division, separates those copies. So it separates those sister chromatids, separates those copies. Okay, and I end up with 4 daughter cells when I'm all done. Right, I have 4 daughter cells down here after the second PMAT. And again, they're all genetically unique. So like we saw with mitosis, seeing this happen under the microscope. So you've got your first division along the top here, reducing the chromosome number. The second division here, separating out the copies. And I end up here at the end with my 4 cells. Okay, so why do this? Well, we mentioned at the very beginning that an advantage to sexual reproduction would be the fact that I'm now creating diversity in the organism. I'm getting genes that are more diverse. Think of if you have siblings, right? Your siblings, they may be a different gender than you. They're going to look a little bit different than you. They're not clones of your parents, right? That's an example of creating diversity there. So sexual reproduction increases genetic diversity, which is a really big deal. When we talk about evolution in the spring, this is definitely going to come back. And it has 3 main ways that it does this. First one, independent assortment. Independent assortment, we can see happening in this picture down here at the bottom. So with independent assortment, remember in metaphase, the chromosomes are lining up along the equator, right? They're lining up along the middle of the cell. Okay, so you can see in this first example of the cells, all the blue ones lined up on one side. So let's say that's dad. All the chromosomes that came from mom are lining up on the other side. Okay, that's not how that has to be. Those chromosome pairs don't care that all the blue ones get on the red side and all the blue ones get on the left side and all the red ones get on the right side. You can see another example of how they could have lined up in the second cell, a second example of how they could have lined up, and a third example, and then another fourth example of how they could have lined up. Those chromosome pairs, this top big pair here does not care what that second pair is doing. Okay, they are only interested in making sure their pair of chromosomes gets lined up on that metaphase, on that equator, on that metaphyseal plate. So they are not paying attention to the other pairs. That's the independent part. The pairs of chromosomes, right? Those homologous chromosomes, those pairs of chromosomes line up independently of one another. So that helps create some diversity. Now that ensures that when you make gametes, right, when your body is making a sperm or an egg cell, that makes sure that when those chromosomes separate into those gametes, you don't have gametes that are only full of chromosomes from your mom and only full of chromosomes from your dad. Okay? Those chromosomes that they gave you get mixed up in making your own gametes. Okay, a second way that they do this is through a process called crossing over. We're going to look at what crossing over is a little bit closer in just a second when I can have a bigger picture on there. Your basic definition is that I'm mixing up alleles, again, across homologous chromosomes. We're going to look at this in just a second. A third way that variety and genetic variation is introduced with meiosis is random fertilization. The body doesn't care which sperm fertilizes which egg. Millions of sperm are released at one time. One egg is released, and only one sperm can fertilize that egg, and it doesn't matter what sperm it is. There's not like a particular sperm that has to fertilize that egg. So random fertilization would also add variety. And like we mentioned, this is going to be our driving force for evolution. So let's look a little bit closer here at crossing over, this third way that we can introduce the genetic variation. We've got random fertilization and independent assortment down already. Crossing over is going to happen during prophase 1. So this is not a mitosis thing. If this was in mitosis, we wouldn't get clones, right? So this is not in mitosis. Okay? When crossing over happens, homologous pairs are going to swap pieces of chromosomes. If you think back to mutations, we talked about a translocation mutation where non-homologous chromosomes swapped pieces of their chromosome. This is not a mutation. This is not that. This is homologous chromosomes. You can see in this picture right here where the little red and blue chromosome are changing over parts of their chromosome. Those parts of the chromosome code for the same thing. Now, let's say they're the parts of the chromosome that code for eye color and hair color. While they might code for different shades of eye color and hair color, one might code for blue eyes and brown hair, and one might code for green eyes and red hair. They're coding for the same trait so they can switch places. So what that does is give us new combinations of traits. It's one of the ways how you can kind of look like a combo of your parents or, a combo of your grandparents and such. So that is where crossing over will come into play. These homologous pairs will switch pieces of their chromosomes during prophase 1. Okay, so that kind of wraps up your basics of meiosis. What's going on, how many divisions, reduction in chromosome number, how we get the variation, why we do it. One of the other things you'll need to be able to do is compare and contrast mitosis and meiosis. So we're going to look at that real quick and we'll be all set. So you have picture format here of comparing, contrasting mitosis, meiosis. You're going to need to be able to put that into summary word format, which hopefully you've maybe already been doing as we've been talking about this, right? So some things that would be a little bit different in mitosis versus meiosis, as well as some things that are the same. Go ahead and take a second and try to do that on your own right now. Do a short comparison, contrast with mitosis, meiosis, and then come back and check what you put together. Okay, so hopefully you got your differences, right? Mitosis 1 division, meiosis 2 divisions, meiosis genetically different daughter cells from both each other as well as the parent to end to end. Mitosis genetically identical to end to end. Daughter cells, mitosis making 2 daughter cells, meiosis making 4. Mitosis being used for growth, repair, asexual reproduction, meiosis being used to make gametes. Mitosis does not have any crossing over, while meiosis does. They do have a few things in common in the sense that they both go through that PMAT form of division. They both copy their DNA first. That'd be kind of about it. Okay? While they seem like they'd be pretty similar, they're very, very different. Okay, so just kind of showing you an example here of how we can create variability through those through these different genetic combinations of meiosis, keeping something similar, but also creating some differences. I forgot, again, my questions reminder slide. Okay, so make sure that you've written down, made notes of your questions, okay, made sure that those are ready to go. All right, and we'll apply all this stuff in class.